Good afternoon, everybody. Everybody wide awake? Ready for a great session? Awesome. It is my distinct pleasure to introduce to you somebody that has a bio that's 5 pages long. And in the interest of time and what he does with his energy and talent, I'll be as brief as possible, but would like to introduce to you Dr. Mark Brantly. He's a professor of medicine and molecular genetics at the University of Florida Department of Medicine. He's also associate director of the University of Florida's Clinical Research Center, and he's the director of the Alpha-1 research program and the Alpha-1 Foundation's DNA and Tissue Bank. Dr. Brantly has been influential in expanding the state of Florida detection program as well as the screening program, and he also coordinates efforts at the Medical University of South Carolina to increase responses to the Alpha-1 coded tested study. And that's a free confidential testing service encouraging people to screen themselves for early detection. But that's all on paper. The real Dr. Brantly is somebody that when a patient calls him or sees him, spends many hours taking care of them. He's somebody that has been in our community for a very, very, very long time. He's the heart and soul of research. He's the heart and soul of screening and detection. And he's the heart and soul of helping us along the way to find a cure for Alpha-1. So without further ado, you're going to have a fantastic session. And Dr. Mark Brantly, you can take the stand.
So I'd like to thank the foundation for inviting me to come and talk. Okay now? How about now? Okay. Maybe I should move it up a little bit. Yeah. Okay, how about that? Is that better now? Terrific. Okay, you might need to turn it just a bit more up. So again, my name is Mark Brantly. It's a pleasure. I see a lot of new faces here, and so that's exciting. The plan is I'm going to take you through Alpha 101. It's going to be a survey. It's not possible to really cover alpha-1 antitrypsin deficiency in a half an hour or an hour or something like that. What I am going to do is give you some of the things that I find that particularly newbies have a hard time getting their head around in the beginning. And it's going to be a lot of genetics and the such that we'll talk about. And then at the end, I'm going to take you through what I say to my patients at the end of a consultation at the University of Florida. And I think that might be a useful thing for you to go through again. So, Angela, thank you for that overly generous introduction in a lot of ways. And so I'd like to go ahead and begin at this point. Our learning objectives are going to be to learn about the clinical and molecular features of alpha-1 antitrypsin deficiency. And then I'm also going to teach you who should be screened for alpha-1 antitrypsin deficiency as well. As you probably know, I'm a genetics professor at the University of Florida. I watch very closely for my audience to see if they're understanding. I also, if they fall asleep, I usually take the laser pointer and sort of shine it on you too. So don't fall asleep during this particular talk from that standpoint. Okay, let's talk about alpha-1 antitrypsin for just a moment. It's a remarkable molecule. It is a remarkable molecule in many ways. It is part of an ancient superfamily of proteins which have multiple functions related to homeostasis of life. And there are multiple genes and proteins that are related to it in this big family that do specialized functions from that standpoint. Alpha-1 antitrypsin has multiple functions, and we're going to discuss about some of those functions as we go through. On your right-hand side is basically a cartoon of what alpha-1 antitrypsin looks like in 3 dimensions. What's stripped away are the sugars that are on it at this point. You can see at the top of that particular thing right there is the reactive site loop which interacts with proteases. But there are so many different areas in the molecule that do other things that we're just discovering even now that are remarkable in controlling it. It is a 52 kilodalton protein, which is about an average size of a protein. It is secreted, and the gene regulatory elements make it so it's responsive to inflammation. So when you get sick and you have inflammation, the amount of alpha-1 normally goes up in your bloodstream to provide additional protection. And we call that an acute phase reactant. It's secreted in abundance, primarily by hepatocytes, but it's also importantly made in several other cells, including alveolar macrophages, which is an important cell in regulation of inflammation in the lung. It's also made in epithelial cells that line the airways. It's also made in gut epithelia as well. So alpha-1 antitrypsin has local functions as well as global functions as it secretes and fills our body spaces from that standpoint. There are at least 150 different variants of alpha-1 antitrypsin. We keep discovering them on a regular basis. In my laboratory, we screen about 140,000 patients a year for alpha-1 antitrypsin deficiency. And so we're coming across new ones all the time and doing DNA sequencing. There's still some more seats back up here if you're interested, and there's a couple of seats down here as well. So, Charlotte Madison, welcome. It's about time you got here. There are about 40 different alleles, and we'll talk about what alleles are in just a moment, that are associated with either low alpha-1 antitrypsin levels or the alpha-1 antitrypsin doesn't work very well. And we'll talk briefly about those. Mutations affect the amount that's secreted and or the function of it. And it looks like that the frequency of Z allele in the population, it's higher than it should be. We usually take mutant genes, usually around 1 in 50,000 people. Obviously, in alpha-1 antitrypsin deficiency, it's around 1 in 2,500 to 1 in 4,000, so it's more frequent. And that suggests some type of selective advantage. There have been a number of proposals about selective advantage. If you've heard the story about sickle cell anemia, where when you have sickle cell, you're more resistant to some of the malaria organisms, we think that there probably has something similar for alpha-1 antitrypsin deficiency. We haven't quite figured out what that particular thing was. But there's also another interesting fact about alpha-1 antitrypsin. Genes can also increase if you have a fertility advantage. And there are several studies that indicate that being a ZZ woman, or maybe even an MZ woman, may be associated with slightly higher fraternal twinning rates. And so if you had a selective advantage in making more babies, that's another way to make a gene go up from that standpoint. In my younger years, I considered making an IUD, was impregnated with alpha-1 antitrypsin. Companies laughed at me, and I didn't go any further with it from that standpoint. So I thought I was going to get rich from that, but I was wrong. So on the right-hand side is a sagittal section, a coronal section of a CT scan from an alpha-1 antitrypsin deficient individual. It's a 51-year-old individual that I used to take care of a while back from that standpoint. As you'll notice, the black areas are where the emphysema is. And this particular gentleman, he has emphysema in different places. The classic presentation is emphysema predominantly in the lower lobe that marches up over time. But in this particular case, this patient has a little bit different. And that's one of the things that we've learned as we've done more intense studies. The classic presentation of alpha-1 antitrypsin, you read in books, you read about that, is not the common presentation. As you guys already know, you're all individuals. You have different exposures and you have different things. So when you realize that in these situations, people are different, and that is an important thing, and I'm going to say this 5 times during this meeting, is that you cannot identify an alpha-1 antitrypsin deficient individual by their clinical features. You cannot. It's a laboratory diagnosis. I want everybody to say that right now. The only way to identify an alpha-1 antitrypsin is by laboratory test, okay? Because there are lots of doctors out there that say, listen, I know an alpha-1 when I see it. The bottom line is Yeah, they know the classic presentation, which represents about 10% of all alphas. Laboratory diagnosis is the only way to do it from that standpoint. Alpha-1 antitrypsin deficiency is associated with a 5-fold drop in alpha-1 antitrypsin in the blood. It's also associated with a amino acid substitution, a single amino acid substitution. So the alpha-1 antitrypsin gene is around 14,000 bases long. It is about 396 amino acids, and one of the bases is changed, and it changes one amino acid in the protein, which makes the changes. And we're going to talk about that in a little bit more depth. Now, why am I going through all these things? Because if you understand some of the biology of alpha-1 antitrypsin deficiency, you will be able to also understand some of the new therapies that are being developed now and some of the clinical trials. So this is your boot camp for understanding what's going on and the things you're going to hear about for the rest of this particular conference. So pay attention, you'll be tested later. Clinical presentations of alpha-1 antitrypsin are quite varied. They can be asthma, just asthma as a presentation. In fact, I have many patients that came to my clinic and said, my doctor treated me with asthma for all these years, and then he realized that I had some emphysema from that standpoint, and that's an important thing. People can have bronchiectasis. Now, bronchiectasis is air tube damage that's very severe. A classic example of bronchiectasis would be cystic fibrosis. 95% of alpha-1 antitrypsin deficient individuals that have lung disease have focal bronchiectasis. So it's an important problem for alpha-1 antitrypsin deficient individuals. A frequent indicator of alpha-1 antitrypsin deficiency is a pneumonia or frequent pneumonias. I can't tell you how many times I have patients come to me and say, Doc, I was fine, and then I got the pneumonia and it never got better. And I'm sure there are lots of people in here that had the same thing happen to them from that standpoint. So that's also an indicator of something. Alpha-1 antitrypsin deficiency is defined by a spectrum of lung disease, which we call chronic obstructive pulmonary disease, which falls into chronic bronchitis, emphysema, some bronchiectasis, fixed airway asthma, and a number of other things from that standpoint. It's also characterized by cirrhosis. Now, I'm not going to tell you a whole lot about cirrhosis because Ginger Clark is going to have a talk right after this, and she's going to tell you everything you need to know about cirrhosis, and we're really learning a lot about that. The last thing I left off my list here is panniculitis. Panniculitis is inflammation of fat. It's a relatively rare manifestation of alpha-1 antitrypsin deficiency, but individuals develop inflammation of the fat, and they can also have ulcers that basically, when the fat starts to inflame bad, it causes ulcers in these individuals from that standpoint. Alpha-1 antitrypsin deficiency can be associated with rapid decline in lung function. And one of the other things, I'm not going to go into a huge amount of detail, is you guys, particularly people that are affected with lung disease, you need to understand the jargon that we as physicians talk about things. FEV1, forced expiratory volume in 1 second, is an incredibly important value for you to understand. It is the one you take a deep breath, blah 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 blah. And the reason why is that doctors pay attention to the absolute number and the percent, and they also, your physician follows the rate of decline in that over time. In other words, how much it changes as it goes down its slope. We make therapeutic decisions based on that. So when you go to a doctor and see somebody and you get your PFTs, write down your PFT numbers, okay? The FEV1 is the most important one for you to remember from that standpoint. And then we know, unfortunately, that it can be associated premature disability and death as well from that standpoint. Now, when I first started in alpha-1 antitrypsin deficiency, the word was if you had alpha-1 antitrypsin deficiency, you're going to die in 5 years. That's 100% not true. Okay, as you well know from that standpoint. People live much longer. And in fact, in a recent study in Sweden, where they compared the Swedish population to individuals that didn't have lung disease when they came into the registry would have alpha-1 antitrypsin deficiency, and they found that alpha-1 antitrypsin deficient individuals actually lived longer than the average Swedish population. Those tended to be the alphas that didn't smoke in that case, and the Swedish population unfortunately smokes quite a bit, so they lived longer. So it is not a death sentence to have alpha-1 antitrypsin deficiency. Can I repeat that one more time? It's not a death sentence. So don't cash in your insurance. You're going to be around a lot longer than you want to. Okay, but it can make you sick. And so we're going to learn about how to keep yourself well in a lot of ways, okay? So what's the problem? It's a relatively common genetic disease as they go. We all rare, common. In the United States, there are at least 75 to 100,000 individuals that are PIZZ. We're estimating that maybe twice as many as we do more and more studies on individuals. But unfortunately, the vast majority of the people have not been diagnosed with alpha-1 antitrypsin deficiency, okay? So we have this model that we've used for years, basically showing that this is our problem right here. And the really important thing is we need to identify those folks, because if we can identify them, we can protect them. We can help them make decisions about not smoking, about many other types of things. And so detection is one of the key things. We can have all the expensive therapies in the world, but if protection prevents most people from developing disease, then that's really where we need to put our money. I mean, we want to prevent people from developing disease in the first place, and I think that's our major goal, and it's one of the foundation's major goals as well from that standpoint. As I mentioned, it's associated with COPD and liver disease and its rapid decline. Asthma is really a very important thing. And one of the things I want to mention is in the population of COPD patients, about 1 in 10 1 in 10 individuals has an abnormal alpha-1 antitrypsin disease gene, okay? What does that mean? That means that of the 16 to 24 million patients with COPD, 10% of them have an abnormal alpha-1 antitrypsin deficiency gene. And 1 in 100 of them will be ZZ. So alpha-1 antitrypsin is the major genetic cause of chronic obstructive pulmonary disease in the United States. We have always said, even if you study COPD, that alpha-1 antitrypsin deficiency, the study of it, is the keys to the kingdom of understanding COPD in a lot of ways. And that's the reason why money is being put into it in many ways from that standpoint. We also believe, and I think some of Charlie Strange's work has really validated that, early diagnosis and preventative care really have important healthcare benefits. So again, early diagnosis, and the diagnosis is made by what method? Yay! Okay. So that was the second time I said that. So the recommendations from a number of august bodies, including the World Health Organization, American Thoracic Society, European Respiratory Society, and the ACCP, which is the Society for the Chest, recommend that all individuals with chronic obstructive pulmonary disease to be checked one time for alpha-1 antitrypsin deficiency, usually by PI typing or genotyping plus an alpha-1 antitrypsin level. All individuals that have asthma or with a non-reversible component should be checked as well. Family members of alpha-1 antitrypsin deficient individuals should be checked as well. And then individuals who have cryptogenic cirrhosis, in other words, it's not obvious as to what the cause is, should be also checked for alpha-1 antitrypsin deficiency. But I want to, again, family members of individuals with COPD definitely need. And I would propose that individuals that have a family history of respiratory disease should also be checked for it as well. So let's talk a little bit about the function of alpha-1 antitrypsin. It has many different functions, and we'll talk a little bit about that as we go forward. But one of the major functions that's classically described is as an anti-protease. It interacts with proteases that basically gobble up collagen and tissue and stuff like that, and including in our lungs. And it basically is inhibited in a 1-to-1. So one enzyme and one alpha-1 antitrypsin molecule basically inhibit each other. And it's a suicide interaction where alpha-1 antitrypsin basically takes out that neutrophil elastase. And it does it in a very interesting way, and the molecular mechanism is here at the top here is basically the alpha-1 antitrypsin reactive site. It acts like bait for the neutrophil elastase. It basically, the neutrophil elastase clamps onto it, and then alpha-1 antitrypsin, like a mousetrap, springs open and stabs that protease and forms a covalent bond and takes it out from that standpoint. That particular neutrophil elastase is no longer able to damage tissue from going forward. Now, this whole concept is engendered in the concept of protease, anti-protease balance from that standpoint. And so we think about, if you imagine just for a minute, if I can take a picture of the lung, If you look in a normal individual, they have a lot of alpha-1 antitrypsin and protection that protects the lung from damage. And they have very little in the way of neutrophil elastase and neutrophils in their lung. Okay? And that's the normal state, and that's where we want to keep. In alpha-1 antitrypsin deficient individuals that have lung disease, and I'm just going to say make sure, have lung disease, then they have more neutrophil elastase and they have less alpha-1 antitrypsin, and that shifts that balance so that there can be proteolytic damage of of the lung from that standpoint. So it's really 2 hits that cause the problem. You can live all your life with very little alpha-1 antitrypsin if you don't encounter Mr. Neutrophil, the guy that brings in the proteolytic enzymes. How do the proteolytic enzymes come in? Well, cigarette smoke is number 1. So, and we talk about risk factors for alpha-1 antitrypsin deficient individuals. Personal smoking is the number 1 risk for causing lung disease in alpha-1 antitrypsin deficient individuals. Second one is passive exposure. We know that alpha-1 antitrypsin deficient individuals that had parents that smoked when they were little have an increased risk of developing liver damage. Occupational things are also an increased risk as well. And those are important ones as well. And then chronic infections of any type or another also play a role as well. So these are some of the drivers that drive lung disease. So many of those things we can avoid. Those are things that we can avoid. We can't really make our parents not smoke when we were first born, but we can do other things from that standpoint and get the word out. Now, as it turns like everything else, I showed you this balance between justice and not justice and that kind of stuff. The lung damage is much more complex than that. And I'd like to introduce this concept because this is how we're developing new therapies for alpha-1 antitrypsin deficient individuals. And so I'd like to say that lung damage in alpha-1 antitrypsin is at least 3 phases. There's an initiation phase, there's a propagation and maintenance phase, and then there are effectors of injury. In other words, the injury comes from that standpoint. And so let me take you briefly through those things. So smoking, we've already talked about. We talked about infections from that standpoint. It also appears that polymers, in other words, abnormal alpha-1 antitrypsin that accumulates, can also attract inflammation as well. And then finally, we're now working on some suggestions that the misfolding of the alpha-1 antitrypsin protein also causes the cell to be injured in a way which sends out signals of inflammation as well. The propagation and maintenance phase is really sort of a dynamic interaction between our innate immune system, which is designed to protect us. And so they get activated, and these pro-inflammatory cells, including neutrophils and many other cells, basically release biological substances like LTB4 and IL-8, which sort of recruit more. So it's this vicious cycle of making more and more cells come into this area and cause damage from that standpoint. And these cells release important things like oxidants, which damage DNA and lipid and cell walls and that kind of stuff. They release proteases that basically gobble up the delicate architecture of the lung. And they make this, and they also release a thing called an alpha-defensin. An alpha-defensin looks like a Klingon bird of prey. And what it's designed to do is to punch holes into cells. Mostly it was designed by the great Megre to punch holes into virus, fungi, and other things. But it's a little unrestricted. It basically punches holes in everything from that standpoint. Interestingly enough, alpha-1 antitrypsin inhibits all of these classes of damaging substances. And that's the reason why alpha-1 antitrypsin is so important in many different stages of lung damage from that standpoint. And so when we think about things, it's definitely really important to think about what it does. And so one of the things is that alpha-1 antitrypsin is a broad-spectrum antiprotease. It inhibits neutrophil defenses and pro-inflammatory, and it acts actually as an antioxidant because it has amino acids that are specifically designed to take out oxidants from that standpoint. So it has multiple manifest functions. And in fact, I'm not going to tell you about some of the really interesting things. We're finding out that it also causes it causes tolerance of inflammation as well. There are some studies that you may have heard about where alpha-1 antitrypsin has been used in different types of organ transplants, and it actually prevents rejection of some of the organs as well. So alpha-1 antitrypsin is this ancient molecule that has multiple functions. I like to think of it as a Swiss Army's knife and has lots of different functions. And I tell you, we're discovering them as we go by from that standpoint. It's a remarkable molecule. I'm definitely one of its #1 fans, and I would like to know everything there is to know about it from that standpoint. So the genesis of lung disease and alpha-1 antitrypsin deficiency at least falls into 3 categories. One is the protease, anti-protease that we talked about. Again, remember why I'm telling you these things. I'm telling you these things because therapeutics are going to hit all of these things. That's what we're going to be looking at from that standpoint. Number 2, polymer are pro-inflammatory, and they may cause more inflammation. And then this unfolded protein response, which also makes cells inflamed, is another area. And so we're designing treatments that hit all of these things and perhaps some more things as we go forward from that standpoint. And when you hear the research trials and that kind of stuff going forward, think about, in Brantly's little Venn diagram, how each of those drugs may play a role in those different things from that standpoint, okay? So this is Genetics 101. I know everybody's been dreading this. It's not that difficult. You know, the 8-year-olds now know all this stuff, so it's time for you to catch up. Okay, so DNA, everybody knows. You know, we have about 1 trillion cells in our body. Each cell has 46 chromosomes, about 2 meters of DNA, 3 billion subunits of ATGC, that are used as the building blocks. We have approximately 30,000 genes. Before the human genome, we thought we had many more genes than that, but we realized there's really not as many. But amazingly, these genes can have multiple functions and do different things, and you can mix and match them and create all sorts of novel proteins. There are far more proteins than there are genes. Isn't that weird? But it's not a one-to-one correlation completely, because they can mix and match. And so the transmission of information goes from the cell, from the DNA, to the gene, to eventually to the protein. And so that's the information. Your DNA basically provides the important information that requires the construction of proteins. And so in this particular case, as you look at this, basically we have these long strings of AGTCAs, which are in a specific order, which encode for information about proteins from that standpoint. And again, it's estimated about 80,000 proteins from this 30,000 genes. Importantly, cells respond to the environment, and they respond in a number of different ways, including affecting the DNA. When we talk about how environment affects the DNA, we call that epigenetics. That's genetics that happens after replication and everything else, and there are many things that influence that, including cigarette smoking, which make basically permanent changes in genetic things which change how proteins are regulated and a number of other things from that standpoint. So what you do now changes your DNA going forward. And that's an important concept. That word is called epigenetics from that standpoint. Now, this is the important information that I want you to try to understand for just a moment. So in each of these things, the way it goes is that the DNA has these what are called codons, and they represent 3 different amino acids. This is what Watson and Crick got the Nobel Prize for many years ago in 1953. Is that we learned that, for instance, if you had a GCA, it codes for an amino acid called an alanine. And alanine is an important amino acid. And you can see that each of these represent different ones from that standpoint. And so if you make a change in the base, you make a change in the amino acid. And the amino acid is the building block of the protein. Everybody get that? Okay. So let's talk about what the consequences of that are. So let me give you an example. Okay, number 1 is that here you have a DNA sequence AATT, and it makes a normal protein. There can be some variation in that, like it changes an A to a T, where it doesn't make any difference at all. makes no change at all in the protein that's made. So that's a silent mutation or a SNP. It doesn't change anything from the disease. But sometimes, if that change is to a C, which actually changes the code sequence and the amino acids different, then that can be associated with the disease, such as sickle cell, alpha-1 antitrypsin deficiency, lung cancer, and a number of other things. So it's those base changes. And so we're going to go back now, and I'm going to put in context about alpha-1 antitrypsin, okay? You're being tested on this, please. Be listening. So here's the big picture. This is chromosome 14, one of our autosomes. And alpha-1 antitrypsin's at the end. The locus is located at the end of the long arm of chromosome 14. And it nests in a cluster called a serpin cluster, serine protease inhibitor cluster. And there are a number of genes in that area that are really related to alpha-1 antitrypsin, including alpha-1 antitrypsin, which we call PI, or protease inhibitor locus from that standpoint. And you can see this is the locus right here, right there. These genes are very, very similar. If you were to line them up on the types of bases they have, they're part of the family of alpha-1 antitrypsin. And that's one of the things that nature does. When it comes on basically a theme they like, they actually just make a little mixes and matches. They make whole new proteins from that standpoint. The locus is associated with a gene right here, which is split. These areas right here are called exons. That's where the information about the protein are. The intervening areas have important information that tells how to assemble the protein and the gene from that standpoint like that. So this spans, these areas right here are the regulatory elements. Genes don't work, all genes don't work in all cells, right? I mean, you wouldn't want to make hemoglobin out of your ears. Right? So there actually is tissue specificity and basically that tissue specificity is based on these elements up here. These are called promoter elements, and they basically say, make this protein in this tissue, but don't make it in the other one from that standpoint. So this is what the alpha-1 antitrypsin gene looks like stretching over. It's got 3 carbohydrate detachment areas. Carbohydrates, so what is a protein? The carbohydrates are added to proteins to make them have a longer half-life, to help them fold in the correct thing, and also to allow them when they lose those, they can say, okay, this protein's old, we need to discard it, we need to get rid of it. That's how we turn over our stuff. For instance, in hemoglobin, when your blood cells start to die and stuff like that, all the carbohydrates fall off it, and they basically go to the elephant place where elephants die from that standpoint. And so it's an important sort of timing signal to say, okay, this protein's worn out, let's go get another one from that standpoint. Let's make another one from that standpoint. So there are a bunch of normal alpha-1 antitrypsin genes. In fact, there are well over 100 alpha-1 antitrypsin. The most common ones are ones like M1, which there are several variants of. And you can notice here, these are just amino acid substitution chains from there. So we have here, I'm listing 4 different types of alpha-1 antitrypsin normal genes, M1, M2, M3, M4, and there was actually M5 and an M6 as well. These genes are normal, but and they don't cause disease from that standpoint. There are a bunch of alpha-1 antitrypsin deficient genes as well. Let me shoot down to the bottom right here. This is a Z mutation right here. It's a glutalyse mutation right here. But you can see there's a number of different mutations. There's the S mutation right there, and there's some really rare ones called improcida and that kind of stuff that are also in here as well. So you hear about these when you're reading these rare genes. Now, 96% of all alpha-1 antitrypsin deficient individuals are ZZ. But then there's this whole rare collection of other mutations that we do, and that's one of my challenges as a geneticist, is to figure out those other ones. And in fact, not only do we have ones that are abnormal from that standpoint, like these, but we also have ones that don't make any alpha-1 antitrypsin at all. Those are called null variants. They basically have a mistake in that genetic sequence that basically messes with basically the translation and transcription of the protein from that standpoint. Those mutations basically sometimes, most of them will encode and say, they're supposed to be reading and saying make, and it says stop, we call that a premature stop codon. So it says, oh, what? I'm supposed to finish this protein, but it's not finished. It's not finished. I need to make the whole thing. But the genetic coding says stop, and so the protein is basically destroyed at that point. And those are called null variants. There are some null variants that are associated with a protein that basically it's made, it is so crappy, it just falls apart inside the cell. We have wonderful quality control information on our cells that basically say, okay, this protein doesn't look right, we're going to destroy that, and they get rid of it. And that's an important conservation thing. We don't want to waste energy in our cells doing those types of things. So there are a number of different mutations associated with these different types of alleles. I've made my career defining these things, and I have had the fun of naming all sorts of funny things like Bonnie Blue and blah, blah, blah from that standpoint. So we name them actually based on the person affected that's the oldest member of a family's birthplace. So that's how we name them in this stem. So get really lucky, you get your name on one of these things. So you notice they're not called Brantly 1, Brantly 2, Brantly 3, Brantly 4. I never wanted a disease named after me from that standpoint. But again, let's go back to one of the most important things. So the Z mutation is by far the one that we really need to understand, and what as scientists we spend the most time on from that standpoint. The Z mutation is a substitution of a glutamic acid for a lysine. Now, all amino acids have a charge. That charge, basically all proteins have a charge. That charge goes from +3 to -3. That sort of defines how proteins are. The environment of the charge tells us whether it is soluble in water. In other words, it's going to be something that dissolves very well in interest tissues, or it doesn't. It's hydrophobic and it doesn't go. It just basically stays where it's going to be. In this particular mutation, the lyse thing, it basically is at the base of this reactive site loop. This is the active site right here, right there. And so it's a hinge area. It's almost like a mouse trap. And so when that area doesn't work right, in this particular case, there's actually a salt bridge on the other side that holds those two things together. On the other side, it's a lysine, and so it pushes apart and allows that big space to open up in the alpha-1 antitrypsin protein, which causes polymerization. And that polymerization is what happens inside of cells that get trapped inside of a liver cell and doesn't allow it to be released. And when it's trapped or it's going slow, our quality control mechanisms start to destroy it very quickly. Now, some of it accumulates, and we think that accumulation causes liver disease, but you have to go to the next lecture to hear more about that from that standpoint. So let's talk about the spectrum of lung disease. As we talked about, people can have normal lung function and a normal CT scan. Now, everybody know what a CT scan is? Okay. It basically cuts you up like a hot dog, and like that when the image is like, and it presents us with a very, very good picture about what's inside your lungs from that standpoint. And so we've learned so much more about that using CT scans. And this is an example of a CT scan of an alpha that we have. And so you can be normal. You can have emphysema, and there are many types of emphysema. There is panacelar emphysema, which is the classic presentation of alpha-1 antitrypsin. That means all the air sacs are affected equally. You can have central lobular, where they affect globules of the lung, and then you have paraseptal, where it goes along septi of the lung from that standpoint. Bronchiectasis is, again, this damage, and this is an example of bronchiectasis, where these big tubes right here, they're open, they lose their hairs, so that, and they're open. Normally airways taper, and you have hairs and you have mucus that trap things. In this particular case, what happens is there is elastin around each of the airways that helps it taper. When you lose that, when that gets damaged by neutrophil elastase and other things, then the airway just dilates open. When it dilates open and it loses those hairs, mucus can accumulate there, and mucus is a very happy growing place for bacteria and such. So the only way to get that mucus out is how? You have to cough. That's right. You have to cough. You have to cough and stuff like that, and you get that nasty sputum out from that standpoint. You know how fast a cough is? 300 miles an hour. 300 miles an hour. So the next time you cough and something lands on somebody, they couldn't get out of the way, I assure you. They couldn't get out of the way from that standpoint. And so it's very important from that standpoint. The other thing I just want to do is to skip down and say that the rate of decline in FE1 is really what defines alpha-1 antitrypsin. And we have epics about when training. We talk about people that have FEV1s from normal to about 80% are predicted, from 80% down to about 35%, and then below 35. And there's different rates of decline in those different areas for people from that standpoint. So again, doctors pay a lot of attention to FEV1 and that kind of stuff. There are a number of good brochures that the foundation can provide you with on how to interpret and understand your PFTs. This is the lingo that you need to understand. To be a good person taking care of your own self, you need to understand what doctors talk about. Now, doctors are responsible telling you, speaking in lay language as well, but if you meet us halfway, things go much better and you really understand things. So learn the lingo that is important from that standpoint. We also know that the lung inflammation begins early in alpha-1 antitrypsin deficient individuals. In the past, I've basically done bronchoalveolar lavage on crazy people that wanted to allow me to do this. And I've actually found, even though they had normal lung function, they actually had some inflammation that was already starting in their lungs. And so we know it starts early, and that means we need to pay close attention to it from that standpoint. The other thing is we think of alpha-1 antitrypsin deficiency as just emphysema, but that's not true. This is a picture of an airway, an abnormal airways from one of my patients. And what you can see is that the airways have been opened up. You can see there's damage right here. This is not supposed to be, they're supposed to be nice and smooth and pinky and everything else. And they're supposed to be small. This has been damaged from that standpoint. And as I mentioned, these alphas have a lot of bronchiectasis. A lot of alphas have what's called a bronchodilator response, which means that they have a response to inhaled bronchodilators. We know that there also are models where alpha-1 antitrypsin actually prevents hyperreactivity. So there was a study done a while back about showing when people are on replacement therapy, they're better able to control their asthma from that standpoint. There's anecdotal evidence that's from augmentation therapy. And then there's a large fraction of alpha-1 antitrypsin individuals that present with asthma as their major feature from that standpoint. I've said that before. This is the only slide I'm going to show you about liver disease and alpha-1 antitrypsin deficiency. That is that the manifestations are cirrhosis and hepatocellular carcinoma from that standpoint. And again, you have to come to Dr. Clark's lecture to hear the next installment of that. So, as I told you, there's a reduction in lifespan for people that have disease. This is old data right now, and that is not true any longer. You used to think the average, the 50% people die at 62 years of age. That's not true anymore. We have had, and this slide is a reminder that we've come a long ways, baby. We have come a long ways. People and alphas are living normal lifespans now. They may be disabled, but they're living normal lifespans. People are not dropping like flies. It seems like it sometimes when you're coming to a meeting like that. But people are living. People are adapting. There are better drugs. We have come a long ways from that standpoint. There are a number of laboratory methods as to how to identify alpha-1 antitrypsin deficient individuals. And how do you identify alpha-1 antitrypsin deficient individuals? Very good. Great, great. Laboratory diagnosis. That's the only way. If you hear a physician say, I know alpha when I see one, just tell them, listen, that's not true. That's not true. It's a laboratory diagnosis, okay? So we have a number of different ways. The most common way we do it is actually by gene-based assays. It's very fast. We can do thousands of them. In my laboratory, I do 500 to 700 of them a day. So in these things, so you can, it's much faster. It's very, very quick, and we can screen a number of them. There's alpha-1 antitrypsin PI typing, sometimes called phenotyping by some people, which uses the technical fact that alpha-1 antitrypsin proteins have different charges, and then if we put these special gels, we allow the charge to change, to basically migrate, and we can see the difference. And I'll show you some pictures of that. PI typing protease inhibitor typing is an important thing to just sort of remember. There are a number of different other ways we can do it. We can do DNA sequencing. We can collect samples from buccal swabs. We can do finger pricks. There are all sorts of ways in which we can do it. And there are a number of assays that are coming down the line that will make the diagnosis easier. There'll be a lot of home test kits and stuff like that that will be coming in over time. Now, this is a schematic of PI typing. protease inhibitor typing. And again, in this case, remember how I told you that it's about a charge? Well, everybody know what pH is, acidity and alkalinity. Well, in this particular case, here is pH 4, and this is pH 5 right here. Alpha-1 antitrypsin migrates the bulk of it at pH 4.5. And so I can take small samples of sera, put it on these gels, which has an enormous electrical current on it. If you touch it, you're shocked. It actually hurts your tits when you do that. It's like so I bet you'll remember that. You won't remember anything else, will you? And so a normal splits up into this banding pattern like that, which is the major ones right here. And you can see over here, ZZ right here, the bands are down closer to 5. And they're also fainter from that standpoint. Now let's look at an MZ individual. They're the normal bands right there, and that's and so you tell the difference from it. This is called PI typing. It's reflex testing. It takes more expertise to do than other types of testing, but in major laboratories that do this on a regular basis, they're all able to do that as well. You can see other variants in here, like for instance, SS right here, which is SS right there. SZ individuals right there, where they have the S bands right here. S bands right here. And so you can see the difference. And here's all the normal variants like that. I'm not going to go into details why the bands are where they are, because I think that's a little too much to do. But remember, PI typing, genotyping, sequencing, these are the ways we identify individuals. The alpha-1 antitrypsin PI type or the genotype dictates what the alpha-1 antitrypsin level is. And so in this particular case, here is QO. QO stands for null. In those individuals, there is no alpha-1 antitrypsin. And therefore, they don't have a level. So null means no, none, 0. An MN individual has a wide range of alpha-1 antitrypsin levels. That's normal. MM is normal. Okay? And that ranges from around 104 milligrams per deciliter to about 270 milligrams per deciliter. This is another nomenclature we use called micromolar sometimes. The normal range is right here. And you can see the ZZ individuals are about 5 times below the normal range. And the MZ individuals actually overlap the normal range. And the importance of that is that you cannot make the diagnosis of MZ by just looking at a level. You have to do genotyping and you have to do PI typing. So a level is insufficient to rule out an alpha-1 antitrypsin abnormal gene. Now, why do we care about MZ individuals? Well, there are a lot more MZ individuals than there are ZZ individuals. Now, the risk of developing lung disease when you're a ZZ is 18 times MM. So it's many-fold higher. But we have 100,000 MZ or ZZ individuals in the United States, or maybe a little bit more than that. We have hundreds, millions of MZ individuals. Now, their risk is not 18 times, but their risk is about 1.5 to 3. So which one has the bigger public health burden? Yeah, the MZs do, really do. And so the Alpha-1 Foundation and a lot of people are turning to trying to understand more about MZs as we go forward, because we realize that the ZZs are important, and we need to understand it, but it's clear that MZ individuals are at greater risk. And why is that important to you guys? Because you have kids. And one of the things we know is pretty clear is if you're MZ and you don't smoke, probably your risk is 0. It's the same as it is for an MN. But if you smoke, your risk is 3 or 4 times higher. So it's very, very important for you to carry that message to your children from that standpoint. This is a typical pedigree, generation 1, 2, and 3. You can see right here. This is an MZ individual mating with an MS individual. And these are the possibilities. But in this type you have basically an individuals that basically are donating their M to here, in this generation right here. So these both individuals are MZ individuals. That's a common presentation. And then in the third generation here, where you're seeing here, you basically have a 25% of being a ZZ individual, a 50% chance of being a carrier, and a 25% chance of being a normal individual in that kind of case. I have patients, families, where everybody that had parents who were MZ were all normal. I have them that are all ZZ, and I have them all as MZ. It's a roll of device. If your mom had 100 children, I guarantee it come out 25, but nobody wants to have 100 children. Yes, ma'am. Okay. Okay, 5 minutes. So genetic counseling is really the most important thing you need when you make the diagnosis. You have to think about that. And doctors, particularly pulmonologists, need to think about family screening from that standpoint. This is the diagnostic algorithm we use in our laboratory at the present time. It's primarily based on a finger stick. We can do other things. And from that finger stick, we can do a lot of family studying. You can have these things taken to your families and stuff like that. And we can make the diagnosis of most genes from that standpoint. So we have to flip the burg. One of the things, as you're in here like that, is this is a genetic disease, okay? And you know, doctors are basically, they want to be heroes. They do. I mean, the reason why we do this crazy life that we oftentimes live in is because we want to be heroes to our patients. But we need a little help from you. We remember patients and diseases based on the patients we have. You remember that person that had, you know, emphysema was young or something like that. We remember those things and we pass them on. You are the best teachers to physicians. You really are. And don't be afraid to ask questions and to advocate and such like that. Doctors want to make you happy. They want to make you better from that standpoint. So speak up. My family needs to be screened, doctor. Can you help us do that? Those types of things. You are a great advocate. And most physicians like that. And the assholes that don't like it, leave them. I mean, leave them. You know, just, I mean, don't stick with them. Because they're more concerned about their pocketbook than they are about your health. And you want to leave them. So we need to flip the burg. And you guys are part of the issue. How can we flip the burg? You help us flip the burg from that standpoint. I'm going to skip this. So alpha-1 antitrypsin is a common rare disease. Smokers die about 20 years before non-smokers do. That's an important thing. Smoking is bad. Smoking is bad. Smoking is bad. Here's the last time I'm going to say it. Laboratory diagnosis is the only way to make the diagnosis, surely, from that standpoint. And the last thing is alpha-1 antitrypsin. I want you to rethink about alpha-1 antitrypsin. It's a conformational disease. It's in the same category as cystic fibrosis, sickle cell anemia, and we have many other things we do. So I'm going to take the last couple of minutes talking to you about basically what I say to patients when we start talking about things from that standpoint. Just a few minutes. So number one is that when I see a patient say, you know, you're a ZZ individual and stuff like that. So number one is you're not going to die. Okay. And the person that determines the most about how you live is you. Important things is you need to avoid infections. That means washing your hands 8 times a day. Staying away from people that have respiratory tract infections. Exercising every single day. And believe me, I know when you're short of breath, it's hard to exercise. But we are an integrated machine. And basically, for most of the people that have lung disease, the only thing broken in their machine is their lungs. And so you have to take everything else out for a drive. When your muscles are efficient, they demand less of your lungs. And so the more efficient your muscles are, the more you can do. You know, I can keep you alive. You know, I can keep you alive. But will it be worth living? That's the question. It is very difficult to exercise when you're short of breath. I know that shortness of breath is a pain equivalent. But if you don't do that, you're not going to be able to make love to your wife or your husband. You're not going to be able to do the things you want. Chronic disease makes your life smaller. You have to push out from that. That's an effort. I have people that I've been taking care of for 20 years with FEV1s of 20% of predicted. I want to repeat this again. That have 1/5 of their lung volume, and they are still going to the gym. They're working out. They work and that kind of stuff. So stop feeling sorry for yourself and get off your ass and do the things you need. You eat right. You need to eat right. You don't eat very much carbs. You eat vegetables. You do what all the gurus tell you to do because they're right. They're right. When you exercise, you have better spirits. When you have better spirits, you're a happier person. You eat right. You take care of yourself. You respect your body from that standpoint. This is the most difficult prescription I put people out on. I say, you have to do that. This is your job. I can't exercise for you. You have to do that job. I can give you the right medicines, I can give you the right advice, and like that. That you are in control of your life. In a lot of ways, you control this whole field because there's only one of me, and there's only one Dr. Strange, and only one Ginger Clark, but there are thousands of you. And so you have to get the message out about alpha-1 antitrypsin deficiency. You have to remind doctors to think about it. You have to remind them that they need to do family screening. You have to remind them about the important things about you. You have all the tools. You have the BFRG. You have these conferences. You have all this information. Use it to your best advantage and live a long time and fool that doctor that said, I think she's going to die or he's going to die. The best thing you can do for a physician to compliment is to outlive me. Okay? So go out there and outlive me, okay? Thank you very much.
Okay, so we have time for 2 questions because Dr. Brantly gave an awesome speech, but we went over a little bit. Anybody have a question? Okay, I just need to wait for the microphone, please, before you ask your question.
My question would be,
Hey, this is DC Young, by the way.
are the graphics you shown today available in a brochure? And if so, where would be able to pick those up?
So the Alpha-1 Foundation has many of my slides on it, and you'll see them all. I just rearrange them sometimes. The same thing is, so you can find them on the websites, on the foundation, with all these different types of slides on them from that standpoint. So you can always look them up. Some of this is from the Barcelona talk that I did a few years ago, so you can see those slides from that one as well. That's posted online as well. Yes, ma'am?
If you're tested and you got your results, your phenotype and everything, and the numbers, which don't matter as much, I understand that, why would a second test show different results?
Okay, so that's a great question. And so why at least in my laboratory, we ask for a second test? The genetic diagnosis of alpha-1 antitrypsin deficiency is something that changes your life and the life of your family forever. Forever. And so the American College of Medical Genetics recommends, when possible, that you get a second test. There can be sample mix-up, there could be something funny about what you did, and that kind of stuff. In the United States, for many, it's free, and there's no reason not to get it from that standpoint. When you have that second test, it's clear from that standpoint. So it's a very good question. It doesn't mean that the test is bad or anything like that, but particularly if you do 100,000 tests a year, guess what? And I test with about 30,000 doctors. They mix it up in their office, or who knows what happens from that standpoint. So it's always important to retest. Okay. Thank you very much.
Thank you, everyone. Dr. Clark will be beginning in 5 minutes, and I will post one of the previous sessions that we have recorded of a presentation similar to this one on our Facebook page. So if you go to Alpha-1 Foundation's Facebook page, it'll be there.